Good afternoon. Thank you for joining the Telo Genomics Investor webinar. Joining us today is Guido Bachler, Chairman of Telo, which is listed on the Venture Exchange with the ticker TELO. Telo provides actionable information that is relevant to medical professionals in the management of cancer and cognitive disease. It is a prognostic and diagnostic company that applies innovative and minimally invasive analysis to improve quality of life and reduce treatment costs. This is a company that I've been following for a number of years, and I think they have a truly groundbreaking platform. I approached management about doing a webinar in conjunction with a fundraiser for the Leukemia and Lymphoma Society of Canada, and I was thrilled that they agreed to participate. Much of the company's focus has been on cancer and recently multiple myeloma, so I think it's a great way to hear about the story and create some awareness about the cause as well. Before we begin, as always, the presentation may contain some forward-looking statements. Disclaimers can be viewed on the company presentation. Following the presentation will be a question and answer period, and questions can be emailed to info@adcap.ca or inputted into the Q&A box below. So thanks for being here. Looking forward to hearing the presentation.
Well, again, thanks, Tina, and the Adelaide Partners team to host this event and for letting us to present Telo Genomics to all of you. I'm excited to introduce Telo Genomics to you today. Some of you may have heard about it. For some of you, it's new. Telo Genomics is in a great position to become a significant player in the multi-billion-dollar diagnostic and liquid market industry. Our forward-looking statement. Let me do brief introductions. At the bottom, you see Dr. Sheriff Louise. Sheriff is actually on the call as well, and we will tag team the Q&A together. Sheriff comes with an extensive experience in molecular diagnostics. He spent a lot of time at University of Manitoba, where we actually work with Telo Genomics technology for many years. He also spent several years at Genome Prairie, where we held increased responsibilities. And then in 2015, he joined Telo Genomics, and since 2018, he's the CEO of the company. We're very excited to have him actually on board. He brings a tremendous amount of information to our team. In addition, the person not here, but I want to give her credit to, is Dr. Sabine May. Sabine May is the founder of Telo Genomics, and she is the leader in the groundbreaking technology development. Myself, I'm Guido Bachler. I'm the chairman, been for about a year. I joined Telo Genomics about 2 years ago as a director. And, you know, the reason why I joined Telo Genomics, it's very simple. I've been in the industry for about 30 years, you know, at Roche, in increasing leadership positions, the executive level in Europe and in US. And then I was leading Singolex, and then most recently was leading the Summer Bio, which is the fastest and the biggest COVID testing laboratory in California. So why did I join Telo Genomics? You know, I've seen a lot of different technologies, and I thought it's very unique, the technology, the way Sabine and the team has put it actually together. It has an extensive reach across many different disease areas, not just cancer, but Alzheimer's and other areas it can be applied as well. It's also proven technology. There are over 160 peer-reviewed publications, as you will see today. It is very clever how they put it together, proprietary technology paired with other technologies that are very well established in the market. That's very important because it will increase your actually market uptake. And quite frankly, there's no other technology I came across that can actually add that additional clinical information like Telo Genomics can do. And on top of that, we have a very strong clinical advisory board, which I will briefly go through here in our next slide. Led by Ken Anderson. Ken is a world-renowned oncologist, heavily focused on multiple myeloma. And Ken has been an incredible good support for Telo Genomics, and he's also helping us on the clinical strategy. And so he's a great partner to have. We also have great relationship with Lawrence Klotz. Lawrence is an oncologist focused on prostate cancer. And you may have seen we've published some really powerful information from him as well. Then Hans Knecht from the McGill Center, focusing Hodgkin's lymphoma, and then Del Druckenberg across a variety of disease areas. So again, you can see we have a great team set up to get this company from the original research stage where Sabine brought it into the clinical market. So the 6 points I will talk about today. The first is genomic instability. We'll talk about genomic instability. We also have a little video. It's very important that you see why we're so unique and why we have such a big opportunity to become a significant player in the liquid biopsy market. Genomic instability is very important to identify whether somebody actually has certain specific cancer or is progressing into a more aggressive cancer form, or how it's actually going to respond to therapy. We use TeloView, which is a software that we're using to analyze the 3D analytics of telomeres, and that's very cool. There's actually nothing out there that I've ever seen that does that, and we'll show you some pictures today. Third point is liquid biopsy market. We're focusing on that. It's a significant market. We'll talk about that in a minute. It's proven. We have significant peer-reviewed publications. We have tested over 3,000 patients. Many smaller companies may have a few hundred patients and may have a few publications. We have a significant amount, which makes us quite different. Opportunities, not just in diagnostic, but prognostic. Also a great opportunity to work in a pharma space for us to be involved in helping better drugs being developed. And last point on the list here is our strong collaboration with the Mayo Clinic, and we'll talk about that as well. We just had a call this week with the principal investigator of our big study that we're doing, and it's the progress we're making is very, very exciting. So liquid biopsy market, it's really probably the fastest growing healthcare market in the US. Not just in the US, I would say globally, but I mean, today the focus is on the US. It's attracting an incredible amount of investors. Just in 2018, over 1.2 billion was raised. Since then, almost every week, you hear about additional fundraising, you hear about other companies acquiring other companies. So it's a very hot market. And we're very excited to be actually, as we move forward and move more in the clinical market, be part of that significant market as well. Next-gen sequencing is a big part of liquid biopsy. You need to know about that as well. We are not replacing liquid biopsy. It's very important that we're seeing ourselves as a partner with with the next-gen sequencing technology, because they're giving you certain information and we're adding to it. So really it is in a beautiful partnership where we can say, okay, next-gen sequencing is providing that clinical information, and we're adding to it to ultimately give a better diagnostic tool for the physicians. And there's some areas, quite frankly, where we know next-gen sequencing just doesn't provide the necessary information. That's where we come in. One of the examples we'll share with you today is predicting the disease progression from smoldering to clinical myeloma. So let me show you a brief video. And I have to apologize, it might be a little bit hacked. It's a big file, but we'll give it a try. So it'll tell you a little bit more about our technology.
Within our cells, safeguarding the chromosomes, are protective structures called telomeres, which maintain the genomic stability needed for healthy cell growth and vitality. Telomeric dysfunction leads to genomic instability, which can trigger disease, including cancer. Within a cell, the three-dimensional arrangement of telomeres, the cellular telomeric profile, is highly predictive of cell health and disease severity. The proprietary TeloView platform can detect the unique telomeric profiles for normal, pre-malignant, and diseased cells. To date, TeloView has been applied to 14 diseases. TeloView harnesses the predictive power of the telomere as a dynamic and universal biomarker. TeloView, genomic analysis for truly personalized medicine.
So just to note that video is also on our website. I encourage you to go up there, look it on again. We also have a lot of peer-reviewed publications up there if you want to learn more about the technology. So for us, there are really several areas that make us very unique in the liquid biopsy space. And this really, as we said before, is a first-in-class diagnostic prognostic tool to analyze genomic instability. Why is this important? And one other point that I think is important that I want to highlight here is that we are actually measuring individual cells. So we do a single cell analysis. Pairing this with measuring genomic instability of a single cell is what makes us so unique. So clearly we use 6 different parameters. We don't choose one, we use 6 different parameters to analyze the tools. The cell are then analyzed with 3D telomere fish. And what we basically do is we label the telomere with the fluorescent tag that can be visualized and then imagined. That creates all the clinical power that we've been talking about before. Stratify patients by progression and the heterogeneity of cancers. Genomic instability profile of a patient is very important. It also predicts response to therapy and guides precision medicine treatment solution. And last but not least is it requires less than 100 cells to actually create a result, which is very unique compared to, for example, next generation sequencing that needs a lot more samples. And as a result, we actually have generally low numbers of sample requirements for performing clinical trials. So let me show you another picture that hopefully highlights to you what Sabine and Sherry actually look in the lab. So here on the left side, you see a distribution of the telomeres. It's kind of similar to what you saw in the video. You see a very unique distribution of those red dots here within the left side, which is a normal cell. When you now look at the right cell, which is a cancerous cell, you see quite a different picture. So you see with the white arrow, you see telomere aggregates, which means they're kind of fusing together. You see with the blue, very short telomeres. So telomeres are shrink in some diseases when they're cancerous. And in other areas, we also see a lot of DNA-free space. So this means the spatial distribution of the telomeres are quite different. And those areas are then basically combined into our technology. So how does an actual test is performed in our lab? How does that actually work? It's a very simple process. So we have 4 steps. First step is sample accessioning. You collect your blood, your urine, whatever you want to perform the test on. Then you perform the 3D telomere immunophage process. You're labeling, specific telomeres and disease-specific proteins with the fluorescent tags. And then you do the 3D imaging acquisition and image processing and the telomere analysis. So what's important here, number 1 and number 2 and number 3 are already done today with different protocols. So nobody does the microscope in the 3 dimensions that we're doing, and the telomere, the fish is slightly modified too as well. But generally, a big part of our process is already currently used in clinical laboratories all around the world. So we're adding a very powerful tool to something that's existing, which generally, increases the adoption rate by clinicians and hospitals. So what are we looking at? Those are the 6 markers I mentioned before. Each is the volume of the overnucleus. We're looking at the length of the telomeres, the number of telomeres per cell. It's very important. Then we look at the aggregates. We saw that before. We're looking at the nuclear position, the distance from the center. And the 6th is an AC ratio, which really tells you how quick the cell is actually dividing, which is a very important parameter for identifying how quick the cancer is actually progressing. Now that we know how this works, I want to show you what we've done on the clinical front so far with this technology. What you see here is a snapshot of all the cancers that Sabine and Sherif's team over the past 20 years have performed studies. It crosses again on the left side, myeloma, Hodgkin's lymphoma, etc. We have prostate cancer. We have data on Alzheimer's disease, on lung cancer. And also on the right side, we have data on breast cancer, melanoma, etc., pancreatic cancer. So there's an incredible opportunity here to focus on. What we as a team have decided to focus on multiple myeloma, because that's the one where we clearly see there's a big clinical need where there are no other tools available. And we will show you a little bit more where we are with that disease progression. So let me talk about multiple myeloma a little bit. Again, it's the lead application for us. because it has a clear unmet clinical need. It's cancer of the white blood cells. It occurs in plasma cells. The symptoms are often pain, fractures, anemia, fatigue, kidney problems, infections. It's unfortunately an incurable cancer. However, you can moderate, through treatment, but it has only about a 52% survival rate over 5 years. So it's still, there's a lot of people dying of multiple myeloma, and we're very excited to be part of that to hopefully reduce that over time. There are over 140,000 cases of multiple myeloma in the US alone in 2020, and the North American market, just for multiple myeloma is supposed to grow to 4.6 billion in 2027. So coming back to my initial introduction, the opportunity of Telo Genomics to be part of the liquid biopsy company, liquid biopsy market is significant. And even just focusing on multiple myeloma is a big opportunity for us to participate. Let's talk about multiple myeloma a little bit more, because I think it's important in the disease progression and see where we are with our studies. So there's different areas when you start, having some initial indications. MGUS is the first kind of signs that somebody may move into an multiple myeloma stage. Again, it's a monoclonal gamma P of undetermined significance. Something is going on in that patient. When it gets more severe is then when someone is identified as having smoldering multiple myeloma. And that is identified with blood tests. And today, physicians identify that and they wait. They don't know whether that patient who has smoldering myeloma is actually going to progress to multiple myeloma or not. And that's where we're coming in. We have done already multiple pilot studies that were able to identify patients that are identified with smoldering, whether they're going to progress or not progress to multiple myeloma. What that means is you can start treatment early and may even prevent or delay, certainly delay, but even prevent somebody from going to multiple myeloma. So that's one area. The second area we're looking at is already somebody has been identified with multiple myeloma. And the retrospective study we're doing is to show the significant differences between multiple myeloma patients who failed initial therapy from those who remain in remission. That's also a very important part. And those are the studies we're actually executing with the Mayo Clinic as we speak. So we announced that by the beginning of 2020, that we're doing a study with the group. There were some delays because of COVID, but we've now really, everything is reopening, thank God. We're moving forward. So again, that's the study number 1, where we basically identify that the patients who progress, which one are not progressing, and the bar at the bottom, just want to highlight this, there is currently no test out there that predicts the transitions from MGUS to SMA or from smoldering to active multiple myeloma. And that's where we're getting in. We're also adding the MGUS in here as well. We're looking at some of those patients as well, but the main focus is the smoldering myeloma. Pretty significant opportunity. And that's the reason why we have institutes like the Mayo Clinic, the NFR, were very excited to actually work with us to identify and perform those studies and ultimately getting into clinical markets so patients can actually use it. The second study is the initial response to the therapy. Briefly mentioned that before. That's also a very, very important study that we're doing with Mayo. So it's really tagged on to the first one. Significant opportunities here. One thing I just want to highlight is that multiple myeloma is a complex disease, and often patients have cocktails of different drugs, and typically it can exceeds of $100,000 a year annual cost. So, if you can help to reduce that cost and help them identify which drug is actually better than the other one, I think we're really good in that area. So now I talked about the technology, the genomic instability, the liquid biopsy market. How do we actually going to bring this into the clinical market? How do we make it possible for patients and physicians to order that test? So there's multiple areas. We announced this as well in our annual meetings, etc. So we see 4 different paths to revenues in providing this information, this tool to partners. The first is license the technology to pharmaceutical companies or research companies, initially to help them to develop better drugs, help identify the right patients who may respond to certain therapies or not. ultimately this could go into companion diagnostic tests. So that's a very important part. And we brought a consultant on board, Craig Mayer, who's had extensive experience in that area, and he's actively now working, helping us to create that opportunity. We also have some discussions about licensed technology to diagnostic laboratories. So it's a way is we have our own lab, or we license the technology to a large lab in the US, for example, who then actually offers the tests to clinicians. The third is what we're exploring quite intensively right now, is having our own CLIA lab based in Toronto, that then actually we could offer in laboratory developed tests to clinicians in the US and also in Canada. And fourth, which is the long-term view, is ultimately we could also develop an in vitro diagnostic test, which requires an FDA approval as a new product. And by doing this, we could then actually really sell it to pretty much any labs we want and don't have a centralized testing. So the next slide shows you a little bit about pricing of some of our other tests out there in the market. So we have this as well on our website. So these are just examples of prices that and reimbursement that some of the other tests in the market have achieved in the liquid biopsy space and others. So you see ranges between $3,500 to $7,000. So there's clearly a space for a telogenomic multi-myeloma test that not just helps identify cancer earlier, but also helps management of therapy to achieve a price in a similar range. Financial summary as of from March 31. So we have currently a $2 million cash in the bank. We have no debt. We have a burn rate of about $85,000 per month. This is trailing 3 months using Q3. The market cap as of Monday, May 24, was 27 million. And the issued shares are 51 million to 51, and then the fully diluted shares, you see those as well. So with this, I want to close out. And again, summarize briefly, it's about genomic instability that Telo Genomics is able to provide. We can analyze it with our 3D structure of the telomeres. We're first in class. There's nothing else out there. We're really going to be part of a significant market. Liquid biopsy, we talked about it. It's a great opportunity. The timing is perfect for us to be here. The collaboration with Mayo is very significant. Again, I think everybody is aware how powerful Mayo Clinic is, not just as a research partner, but ultimately as a potential first client to start leveraging our technology. It's a well-established technology, and we have significant opportunities, again, in the diagnostic, prognostic products, pharmaceutical partnerships, and academic collaborations. So with that, I think we have some time left for Q&A.
Yeah, that's great. Well, thank you for the presentation. If anyone has any questions, you can enter them into the Q&A box below. Why don't we start with your involvement in the company? You're an industry veteran, obviously. What about the company did you find so unique, and what's your view on the company's potential in the industry?
Yeah, well, Tina, that's a great question. So I think as I started off, when I started doing research on the company, I was highly impressed, first of all, by Sabine May's research. She raised 25 million non-dilutive funding. I think Sabine May has probably the most advanced microscopy lab in North America. I mean, it's amazing what she has. And so, talking to her and learning about the technology, learning about the potential, understanding liquid biopsy, understanding of the technology, it was very clear to me, this is a hidden gem here, in Manitoba, Winnipeg, that is, exciting. So again, quite frankly, Sabine pulled me in, with her strong knowledge, and then, meeting Sheriff and the team, and, that's convinced me, in one other, maybe a little note is, I talked to Ken Anderson as well. And, he's a believer in the technology, and Ken Anderson knows a lot of technologies out there. And his trust in Sabine's technology was also another point to me that was, this is unique, this is real. Let's help this company to get into the clinical market.
And the company's done work on a number of cancers and Alzheimer's. Why did you choose multiple myeloma as your initial or lead application?
Yeah, that's another good question. We have, as you said, sometimes you have too many options. You got to figure out which one is the right one to choose. Multiple myeloma for us, clear. There's a significant unmet clinical need, right? As we had seen some initial studies from Sabine, we looked at that. It's also not a very crowded field, right? So there's not, when you look at other cancer markers, you are one of 100 or 20 trying to do something unique. Multiple myeloma is a little bit a unique market still, and so that's, again, Tina, there were several reasons, but those are the ones that ultimately made us decisions with the relationship with Mayo as well, considering.
So how did Mayo get involved in the company to begin with?
Relationship with Ken Anderson and Sabine May from the past. They've done work together in the past, and so that's how this all started off. So and that's why I think it's, Ken is very busy, as you can imagine. Everybody wants to work with him, and we can call him, and he helps us all the time. So that relationship started it, and probably helped also by we doing multiple myeloma.
Can you go into a bit more depth about how the diagnostic will inform treatment and modulate outcomes for multiple myeloma?
Yeah, so I think I'll do one more, and then I'll let Sherry add some points as well. So I think that there is no clear test today to identify whether a patient that is identified with smoldering is going to progress or not. With our tool to be able to identify now with the high sensitivity and specificity that you will progress or you will not progress, in one hand gives the patient the peace of mind because, it doesn't have to worry as much and may has to be monitored less frequently, right? But somebody who's actually identified as a high risk, the oncologist may start treatment earlier on, and studies are coming out to start showing that if you actually treat early, you have a better outcome. So I think that's an important part of just multiple myeloma where we can have a significant clinical impact.
We'll get Sharif to come on as well to go through the Q&A. Hi, how are you?
Good. How are you? Nice to see you, Tina.
Good to see you too. Yeah, exciting news that you guys have put out recently as well. So in regards to multiple myeloma, can you talk about the most recent results that you've seen and the significance of them?
For sure. We're, as Guido alluded, we are now in an ongoing studies with the Mayo Clinic. Recently, there was a publication, just came out last month, and it included several groups of patients. Smoldering myeloma was one of these, and we saw, as the publication says, like very high significance across all the parameters that we measure between stable smoldering and progressing smoldering. And to the question how this can influence the treatment, there is a strong evidence in the myeloma community, in the clinical community. It's a very close community. To start treating smoldering patients, high-risk smoldering patients, if they can pinpoint them. But to Guido's point earlier, there is no test that can pinpoint and say, among this group of smoldering patients, these 5 are going to progress versus these 5 are going to be stable. If we can give them this piece of information, they will start treating these patients right away, and that will delay their progress to multiple myeloma. This is a significant change in the management of this disease.
You said there's no kind of test that does that currently. Can you touch on the competition a little bit and what you're seeing in the market right now?
For sure, and Guido alluded to that as well. Like, it's not a busy space. There are few clinical risk factors that now clinicians, out of being desperate for a clear-cut evidence, they look at them, and these are like standard old-style protein blood markers that they can try to make educated decisions based on these little tools. If that protein is up, if this number of cells is higher, the probability might be a lot higher that patient will progress. But there is no really clear-cut test on the market today that can say this is a high-risk smoldering, and I'm going to start treating it now.
And you mentioned that the multiple myeloma data was published recently. I think it was in the scientific journal. What has the feedback been like so far from the medical community?
I think it's amazing. A lot of traction, and maybe I would say On a weekly basis now, we are talking to different clinicians and key opinion leaders. Certainly, this publication is going to open for us more important collaborations with key opinion leaders in the space of multiple myeloma, different stages of multiple myeloma, for sure.
And you touched on some of the paths to revenue in the presentation. Can you expand on some of those paths, what stage you're at in terms of licensing agreements? And you mentioned potential for a CLIA lab as well. Where are you in that development process?
We're starting to explore the path to have a CLIA lab. I think it's important for us in any case and in any path to revenue in any collaboration. We run world level and state of art operations in our laboratory, but having the designation is going to be an assurance for that. So we so much look forward into it. We recently also retained a consultant to help us out with this, with the heavy experience in this area. So I'm having the CLIA designation is gonna shorten the path to revenue by being able to offer laboratory developed tests to the market as soon as we complete our clinical studies. And I know maybe Guido would want to add to that point as well.
Yeah, no, I mean, totally agree. So clearly, this is a process. So the CLIA lab is an important part. I actually personally built two CLIA labs from scratch. So Sherif and I working with somebody to help us think through the process to getting it established in Canada. It'll take some time. What are the requirements, etc. So this is a big part. And on the relationship, it's also important, Greg, that it helps us. He's been doing this, and I actually know Greg from the past. We've done some work together. When I was running Singlex, we had protein platforms that we were leveraging, also selling to pharma companies. So it's a well-established process, and Greg is really helping us to have inroads, with pharma companies. For example, there are also diagnostic companies like Roche has a big lab, for example, where they offer fish technology to pharma companies all around the world, and so is Siemens, and there are other ones. So we're starting to kind of connect with some of those diagnostic companies and starting to explain our technology and moving forward with partnership. But it's too early to talk about anything more than that.
Right. So there are two studies that are ongoing with multiple myeloma. What do you think the path to commercialization is? How long do you think it'll take to get there?
Yeah, I don't think, we can specifically talk about the time frame because we need to look at the results. We need to make sure we're comfortable with everything. We expect them to be really looking very good, but then we have to have the publication be published, etc. So clearly, we understand the timing, but we need to do some more work on that before we go public with it.
And multiple myeloma is obviously not the only study that the company's done. Can you discuss some of the historical findings and research with the other diseases that the company's done testing on?
That's a perfect question for Sherif.
For sure. We completed the clinical study that we presented actually in ASH. It's a hematological disorder on Hodgkin's lymphoma. This is a completed study. Away from that study also, in collaboration with Sabina Mai's lab in Manitoba, she is using the company's technology, and we have very strong publications of completed studies on Alzheimer's disease, 2 independent cohorts, both of them published. We have a very strong publication on a prostate cancer cohort with very high prediction power, a negative predictive value, a positive predictive value that would beat everything actually competing on the market. This is all public domain. These are all published papers. So we're so proud of the prostate cancer study, about the Alzheimer's study, about the Hodgkin's study.
So if you're an investor looking at the company, where's the best place to find some of the data on Alzheimer's, prostate, Hodgkin's?
Well, I think, as Guido mentioned in the presentation, collectively we identified that multiple myeloma is really the lead indication and is really the strong and the powerful proof of principle for this company. And we wanted to make sure that we drive that test to the clinic. The pipeline, as I said, those are the key players, but for sure the company now want to focus on driving these tests to the clinic.
And can you touch on the patent portfolio as well?
For sure. We have currently 17 patents and pending patent applications, and that term does not give it its right, because 16 of these are already granted and only one pending application is there. We're covered with two sets or two groups of patents that govern our rights, our intellectual property in the core technology, what we do, as Guido explained it before early in the presentation, and another group of patents that govern disease-specific intellectual property. We're covered in Canada and the US and in Europe.
What do you see are the biggest risks for the company going forward?
Guido, do you want to?
Yeah, so I think we have done a lot of pilot studies, right? We know where the data is, where the clinical data is. So I would say the risk is the timing getting the study finished, executed, and getting into the market. So I think that's, to me, probably the biggest risk. The technology works, it's been proven. One of the reasons I joined, because I know this technology works. The way it's uniquely positioned in the liquid biopsy space, it occupies a very specific area, and we see this. So generally, that's what I see the biggest risk.
And turning like risks gonna come, but it's the ability to mitigate risks, and I think we demonstrated through the COVID time our resilience to mitigate all these risks.
And just looking at the balance sheet for a minute, can you talk about, you mentioned you had $2 million cash, I think $85,000 a month in burn rate. I think you had some warrants that were exercised recently as well. Can you just give an overview of the budget?
For sure. We had two waves of warrant exercises. One was in November 2020, and that returned a revenue over $1 million for the company, and another wave of warrant exercise, and that was in February. That was in the ballpark of around $600,000, and these warrants are all related to the latest private placement that we had in November 2019, and it just shows that our investors renewing their trust in the company, satisfied with the progress, and as I said, with the resilience we demonstrated, the budget responsibility we demonstrated across COVID, our burn rate as committed. So I think it was very thrilling to see the investors coming forth and exercising their warrants.
And is the shareholder base predominantly retail, or how is it split between retail and institutions?
Well, it's kind of a split. It's kind of a split. I wouldn't want to elaborate more than that, but it's kind of a split.
Okay. There's a question from the audience as well that was just asking about plans for investor outreach.
Guido, you want to handle that, or want you to?
Yeah, you can. You go ahead.
Sure. We're open for business, and we're always happy to chat with investors. We attend conferences, investors conferences, and we talk to everyone there. And when the right time comes, we're happy to the right time, the right opportunities. We're always happy to go serious in negotiating capital raises. But as you said, like our cash position is now good. We are very responsible when it comes to budgets, so we are comfortable.
Okay, great. If anyone has any other questions, you can feel free to enter them into the Q&A box below. Otherwise, I'll leave you with, if you could summarize what the next year ahead will look like for the company and some of the catalysts to watch out for.
Guido?
Sorry, you want to just start, Sheriff?
This is a summary for the next year outlook.
Right. Yeah. So, sorry, I didn't connect that. So basically, Tina, the outlook really is to focus on the study with multiple myeloma. driving that clinical study forward, addressing the development of the commercial strategy with the CLIA lab. We'll also hire some an outside group from a PR point of view, so that addresses kind of the investor outreach question from before. So clearly, as Sheriff said, focus. We're going to focus mostly on multiple myeloma. We are, of course, opportunistic, exploring additional studies, as we briefly talked about, that we're building out and expanding our clinical advisory board. Certainly focus on multiple myeloma, but we may do another study, depending on how things are going. But the key message is our focus is multiple myeloma, and we're opportunistic with other studies that come along.
That's great. Well, thank you so much. Appreciate the update, and congratulations on all the success you've had so far. Obviously, some fantastic data coming out of the company. So looking forward to keeping on following these trials.
Thank you so much, Tina, again, for letting us present today.